Good evening, everybody. We are getting close to the year, and obviously you may have noticed who follow us that we had two important announcements. One on last Thursday morning, post our Independence Day, and the second one this morning, both dealing with our lead product, bexmarilimab, or bex like we call it. And our purpose is really to reprogram myeloid cells that are born in the bone marrow, regulate a number of things in us, keep us clean, and then also participate in preventing several types of the diseases. With this, obviously, I want to show also our disclaimer because I will be telling some of the things which are related to the future activities. So, bexmarilimab, the mode of action we have known already for a while. It's really to reprogram these phagocyzing capacity cells in us who are, in often cases, our janitors. For example, they clean our brains, but they also participate in the defense system. They can be immune suppressive, and that is the capacity which is really needed, for example, during the pregnancy, when the embryo has to be protected against mother's immune system. Without these cells, embryo would be resected rather soon when they start to grow. Cancer has taken advantage of this, and they also hide, by a similar way, our defense system. And when they do that, there is no recognition that they actually are dangerous to us, allowing them to grow first locally and then starting the metastasis. And obviously, the metastasis is the problem which actually are killing us. Unfortunately, there are still several cancer types that we have difficulties to treat with at the moment. When we expose these immune suppressive cells against or with the bexmarilimab humanized antibody, we reprogram them, and now they become immune activators. And you can see that in this lower panel of the picture. It will activate the Th1, the very primary immune activation system, and then if that becomes active against any of the cancer antigens, you actually generate your immunity against those, and then you should be much better off in fighting against the cancer. And obviously that type of the situation is already demonstrated with the PD-1 blockade, where the activation is taking place on those cells which already are in those individuals. Unfortunately, it's only one portion of the population that actually get the help. So something additional is needed. And we believe that this reactivation of our innate immunity is the key thing really to expand the immuno-oncological methods really to help these cancer patients. We just got this first MATINS trial data with the advanced solid tumors accepted in the very prestigious journal, Cell Series, that became online on last Wednesday. And in that one, we have studied some 200 patients, shown that the treatment is extremely safe. There is no really serious adverse events involved when exposing patients to this antibody. We then also have learned that the activation of this conversion from immune suppressive macrophages into immune activators happens within the tumors of those patients, and that is based on looking at these tumors post pre-treatment and post-treatment periods. And that is shown really in a macrophages that are located within the tumor. That is important thing, that because if that doesn't happen, then we cannot help really those patients. We also learned that if you have a low baseline immune reaction in those patients, those are the ones we can help. Those patients are often called, or the tumors are often called cold, meaning that there are no immune cells. And those are the ones that actually benefit of this treatment. And that can be shown in the interferon gamma, one of the major mediators of immunity increasement. And when that happens, you have a survival benefit, which is then shown in the middle of those lower panels. You have a disease response which is really rather significant among those patients. And as you can see, these are last line patients, and we still have a response up to 40% in some of the cancer types. So very crucial finding to move forward. But then if you look at these hematological cancer types, there are many of them, but we obviously are very interested in those where the origin coming from the same myeloid cells as those monocytes and macrophages are. the simple interest here is that these cells now carry also the clever-1, the same target we are targeting on the macrophages. We have learned from ex vivo settings that we can again activate the environment against these cells because in the several settings they increase the antigen presentation, and obviously that's the first activity you can see within those environments. Then on top of it, we also shut off the energy production in those blasts, the cancer cells, and that decline their viability. So two mode of actions in one antibody treatment. And obviously, if you believe that this is true, we should have really fantastic results. And I'm not really showing more of the experimental data. I just want to demonstrate that this is not only related to these myeloid cells. Clever-1 is expressed in some of the other hematological malignant cells. So obviously the use could be very much larger than just focusing on them. But especially those myelodifferentiated syndrome patients, which are called MDS, if they do not respond to the current treatment, they have really poor outcome, and really mean poor. The survival rate, 90% of them will die within the first 2 years of the recognition of this condition. And this high-risk MDS patient population is really significant also. This is the distribution of those patients when they currently go over during the treatment when we try to rescue them. Only very small proportion are lucky enough to get into the bone marrow transplantation and survive. That is considered today perhaps the only long-term treatment that can help these patients. So from these other lines of treatments, they then end up in this refractory population where there are really no options for the patients. And this high-risk MDS population alone is roughly 90,000 patients in US and 5 biggest countries in Europe. So very significant population just alone. Our first study, BEXMAP, which we have been now doing a year and a half, it's really phase 1, phase 2. Typical starting point is to dose the patients with different dosing levels. Those we obtained from our MATINS trial, and we hit the right levels also with these patients. Dosing being 1, 3, and 6 mg per kg. In here, 1 week interval. In the MATINS trial, we started with the 3-week interval, but we have decided to increase that with here because of the high number of the cancer cells, those blasts in those patients. The results I will share with you rather soon, but we can also now say that it was already very well tolerated within these patients as well. So we have now passed the first part and have already initiated the second phase 2 part where we follow the FDA guidance, where we need to really finally test 2 different dose levels and then select the best safety plus efficacy candidate dosing level and then move on to the pivotal part. That part will not be very large in size. It's either 20 to 32 patients, and it's randomized between these 2 dosing levels, and that's very soon starting to go. And obviously that will be the focus for us in 2024. Well, I already said that within the MATINS trial, this is very safe, but it's also safe with these hematological malignancies. One of the visible things you have observed with the leukemia patients is the baldness. People are losing their hair, and that is the reason that the treatments are usually very toxic to the patients and they lose them. None of our patients didn't need to go through that type of the situations. There are very few adverse events which are related to the bexmarilimab itself. And obviously this has improved our clinical capacity because the clinicians are extremely excited having a drug candidate for the cancer patients which is not causing a severe adverse events. Some of the ones that have been taken place, they can be really also treated with the steroid and patients can come back to the trial. So obviously that is a extremely nice and good thing. Then we also have shown that within these patients who have been treated with the BEX, you really activate the immune system. On the left-hand side of this picture, you can see the antigen presentation capacity growing while the dose levels are increasing, and on the right-hand side you can see the accumulation of these immune cells into the bone marrow as predicted. So this is always nice to see that the mode of action is exactly what we have described. So then on top of it, we obviously hope that now when the antibody is administered, they actually attack the cancer cells and their energy production goes down and they die. This is the current extended swimming pool showing that we have a number of patients, roughly half of the population we have treated within the myeloid malignancies responding to the treatment. And that response can be seen either in the reduction of these cancer cells in the bone marrow and the circulation, or with the improved blood values like hemoglobin or white blood cells. Those are the really important things, because often the patients really do not have any blood substitutes left, and they need to substitute in order to really maintain the capacity to prevent the infections. So very good progress, which is now long-lasting and very interesting one. And if you look these more carefully, we notice that the two-type MDS type of the diseases, we had extremely satisfactory response rates. 5 out of 5 with this high risk MDS, and then also 5 out of 5 with the HMA failed MDS. People don't necessarily like to say that we had a 100% response rate, but that the numbers are, but this is a very promising. If you look at normally the survival time with these patients is only from 4 to 6 months, and we have much more extended time period, and many of these patients have lost these blasts completely. Works exactly like we have really aiming to get the drug to work. One point I also want to say that you can see that there's already one transplantation patient here went further on and it's out of the trial. And then we have several P53 mutation carrying patients, and that is very dangerous to these patients when they develop these, and there has been really no response rates with the current treatments. So 4 out of 7 patients, if we have a response, really carry this type of the mutants as well. This is a extremely exciting situation to be, and this is just to describe the same thing with another way. Here is the photo graph showing that the cancer cells, immune blasts, are really disappearing from the circulation while they are treated. There is one patient here who actually got the bexmarilimab maybe too late. It was converted into the acute myeloid leukemia, and there's really nothing there. They are very aggressive form after this period of times. The patients have been in the previous treatments. We couldn't help that patient at this stage. But you can see a nice response, 9 out of the 10 patients. So in the summary of this part, humanized antibody really priming the activation of the immune system, very safe, definitely showing a efficacy, which is extremely interesting. And obviously we have now converted our focus from other leukemia forms into MDS, because it looks like it's really the fastest way to the market at this point. And that is what I'd like to really discuss first. We have now completed the phase 1 part, got the dosing levels, have now really started the preparation to initiate the phase 2 that we will be carrying out with the 2 different concentrations, 3 mg per kg and 6 mg per kg every week. And when that is done, we also discuss this with the regulators, especially with the FDA, who have been giving already us very significant feedback, and then pick up the final dose and move on to the pivotal part. As I said earlier, this part 2, phase 2 part, is only maximum of 32 patients. That allows us to select the right dosing level to move on to the pivotal part. Usually in the setting like this, where there is nothing to the patients, we don't need to do really carry out trial with the 200 or 300 or 400 patients. It will be less than 100 totally. And obviously that is our wish, and at the same time discuss if we can really get the accelerated approval to start with, and then make a confirmatory trial at the same time. If FDA or EMA do not agree with that one, we can also go directly to the randomized trial and to complete that part. We anticipate that we can actually make a biological application already in 2025 for this indication. You may recall how about AML, because that is also the target. We may need to do for AML a similar Project Optimus part in other ways, test 2 different dosing levels, and the reason is very simple. AML patients carry much higher number of these cells in their circulation, so it may be really reasonable thing to test that and then let FDA to decide how to move on with that part of the trial. So roughly summarizing this, patients do not have that much of help from the current treatments. Half of them already responded in these early settings of the trial. Almost 100% or maybe 100% response rate with these MDS patients, both types. And the response is very deep, meaning that the blasts are really disappearing. We either have a complete response or complete remission, or then bone marrow remission. Both are considered as a really good progress in the patients who have been carrying these blasts for a while already. Then aiming really the accelerated approval, including the discussion with the regulators, and then having a situation where we can actually one day say that doing this, we will be on the market. Having said that, we had really poster presentation yesterday in San Diego. That is American Society for Hematological Conference. I can tell you that the crowd around that poster was a really significant. We saw that already earlier in the similar European conference, and obviously we have a number of different commercial meetings there as well. So I can just say that this will boost our partner discussions, and you might be interested in learning more about that, as I will do, and hopefully sometime during the first half next year we have something concrete really to offer. Meanwhile, we will really initiate the phase 2 and then hopefully get the full data readout for that one already on the last quarter next year, but hopefully even earlier. The current activity also includes the expansion of our clinical sites in US. We have had 2 sites in their City of Hope close to LA, and then MD Anderson in Houston, but there will be additional sites in Yale and North Carolina, in Fred Hutchinson in Seattle, and hopefully some other ones. And very nice thing is that we actually have a lineup of sites who actually would like to join the trial, just due to the fact that safe, early efficacy, and there's really nothing for these patients at this point. So I was hoping to have a slide at the end to show the crowd around the poster, but I didn't get that. I just heard that there is a significant really crowd. But all of this, really want to thank you for your time, and I'm happy to then also have additional questions from the audience, whatever they are. Leave that.
Thank you, Markku, and please have a seat. Okay, we have plenty of questions here from the chat, so let's start. So the first question, the person first congratulates you on the new data, and the question is on the commercialization strategy of bexmarilimab. So what do you see is the best way to move forward? For example, licensing for specific indications, or licensing to one or several companies, or what is the optimal way for you to move forward to get the best benefit from Bex?
Looking at the feedback which we already have, it's becoming rather evident that we want to have one that actually controls everything. So it will be global license, all the indications. We have initiated a competitive auction process already, so it's moving on, but obviously there are a number of activities we have to take, and we'll really hopefully get further information on the first quarter how well it's actually progressing. There is a significant need, and every company who actually have been in this space understand the value of the early efficacy, what you can see from those patients, because there's really nothing for them at the moment.
You have been doing the Martin's trial previously, now focusing on BEXMAB. So one question is that kind of the need for partnership is and has been very clear. So why haven't you partnered yet? Is it because of lack of good deals or why?
Well, yeah, I mean, honestly, I'm really aiming high because this is really the mode of action that is really needed to really get the immune activation taken place in those patients who already have been declined or shut off by the cancer. The PD-1 market is very interesting because, as you know, there are some 10 different products on the market at the moment. Just recently, we learned 3 major phase 3 trials failed with Merck. It's the point where they need to understand what kind of settings they would like to do with the bexmarilimab. And obviously, we are providing additional tools. For example, we can now, or we do have a companion diagnostic assay to look at what are the intratumoral Clever-1 presence in those patients who are already negative for the PD-1 treatment. So combining that already in the setting that also the other side will appreciate and understand, that has taken some time, but we are getting there as well.
Thank you. So now you're focusing in BEXMAB phase 2, you're focusing on those MDS patients who have failed
Correct.
the HMA treatment.
Correct.
And so firstly, why are you moving forward with this cohort? And secondly, how about the first-line MDS patients and the AML patients? Are they coming along too?
Obviously, the refractory population, it's the one that actually the highest need. And as there is no really treatment for them, we are still hoping that we can make a single-arm study with that population, meaning that there is no really comparator arm. However, if FDA says that you need to do that, then it's doctor's choice that they pick up, and we can randomize that with 2-to-1, having a BEX treatment in 2-to-1 with the standard of care. And then run it that way. It will a little bit delay maybe the startup, but again, then you have to have a IDMC study committee that's looking carefully the separation of those patients, because if this is so devastating condition that your predicted lifetime is only 4 months, you can't put those patients in that arm. And if it shows up at the very early on, then we go to the single arm trial to continue it. This we need to discuss with the regulators later on, but that is the reason that we believe that the fastest way to the market is really this refractory MDS.
Okay. There's a question on applying for preliminary approval after phase 2. I think you already touched this in your presentation.
It's possible, but again, that has to be discussed, and it obviously it depends on the efficacy level of that. If you think about the current survival rate for those refractory MDAs, within the first 2 years, it's no more than 10%, most likely less than 10%. There are different publications indicating that. We have had tremendous kind of a response rate, and if that holds up, why we need to run a comparative arm if they are so separated already from each other. So we have to carefully follow the opportunity, and that's what we are doing.
Okay. So what are your plans on utilizing breakthrough programs or similar kind of fast-track programs from here on?
Our medical advisors and writers are really busy. They don't have a Christmas holiday break.
I see. You clearly are too small and underfunded for all the current possibilities you have in your pipeline. How are you going to maximize the potential of current pipeline?
Well, that also goes back to those partner candidates. We need to make sure that they do not prevent the expansion of the program. So they have to commit themselves. And that's part of the discussions we need to go with them. And that has actually heavy impact on the selection of the right partner. Having said that, obviously with these results and some hopefully additional ones, we have been able to finance us the this year, which is not very easy because the markets are not very good, but yet we have been able to really move on. And with this careful focus that we now push forward this BEXMAP study to the point that we actually can really see that it's doable really to get it to the market. We then meanwhile make some adjustments to our BEX combo study, which we have said that we have been already discussed with the regulators, and then get that ready, and hopefully that it's good enough plan for the partner candidates to accept that as well.
Okay. So you have previously talked about the investigator-initiated BEX-lung study for non-small cell lung cancer, and you have also some plans for the NLSC in the BEX combo trial. So how do these play together, and what are your plans concerning lung cancer?
You're absolutely right, and I'm a little bit disappointed that we couldn't get that started in San Antonio, in Texas. But now we actually have several PI, principal investigator-driven studies planned, and obviously they do not cost us that much as the trials we are sponsoring. But the problem is that you have difficulties to push them forward. So this is now the compromise where we try to balance with our major target where we are the sponsor, and then the additional test trials where we can actually get information and learn from there how we actually will like to build up the last line refractory population trials.
Okay, and there's a question on biomarkers for the BEX combo study. So you have talked about interferon gamma as a possible marker, but what does it look like now for the BEX combo?
We already know that higher the intracellular Clever-1 immunohistological staining is a poor outcome of the patient cancer will be. So there is a strong association between those items, and obviously the companion diagnostics can be built between or using the immunohistological staining. That is a different antibody than bexmarilimab, because bexmarilimab is a function blocking antibody and cannot use in staining systems. So there is a separate antibody for that. And that has been adapted already for the commercial diagnostic instrumentation in hospitals. So I believe that is something that we will build into BEX combo studies. But then there are some other ones. There is a way of looking at the interferon gamma signature baseline, and we are involved in those developments, but they are not standardized yet. So we may need to do some additional things. And then we have some additional thoughts as well, how we actually going to combine. So that would be that from that refractory solid cancer patient population, we actually would like to go in first by picking them up and then running the trial and then expanding later on.
Thank you very much for the interview.
Thank you.
And next up we have Herantis Pharma and Antti Vuolanto.